Our days at sea, we have presentations by our university study leaders going on at regular intervals, keeping us occupied and entertained.
For me, it was wonderful to find out there was so much activity incorporated, and essentially it was at your own pace and at the level you wanted.
We encountered an iceberg that was so large, we had to divert our course. We had to actually go around the iceberg.
It took us so much time to get around that on our way to the Antarctic Peninsula that we just went straight to the peninsula.
From all the way left, all the way to the right, it was just iceberg for us, as far as you could see.
And between the Worldwide Quest team and the ship's team, they also made it fun. It was all part of the experience of being on an expedition. You had to expect those kind of changes, and it became part of the adventure.
Well, hello there. Good morning, or perhaps good afternoon, depending on where you're joining us from today. My name is Justin Peter, and I'm today's host for your presentation. I am also one of the owners of Worldwide Quest, a small travel company based here in Toronto. And I'll take a moment to tell you a little bit more about us in just a minute or so. First, I wanted to say, in terms of technical issues you may be facing, if you can see me, but you cannot see a slide, or vice versa, you should be able to see my face right now, as well as a slide with the title of today's presentation. If you don't see that slide, below my head, below the window with my head, there is the gray bar, sort of the box of the window, and you might see three little parallel horizontal lines, sort of like a hamburger. If you click on that, the slide with the actual presentation should pop up for you. So just in case you're facing that challenge, if there is some reason you need to leave today's presentation, be aware that our presentation is being recorded today and will be shared with you afterwards by email with a link to your email, the one that you used to register. So if you do need to leave us early, know that you can return and you will see the entire presentation. All right, today's presentation should be taking about 45 minutes. We'll have a presentation about 35 minutes or so in length, and that will be followed by a question period with today's speaker, Philippe Tortell. And on that note, in your GoToWebinar console, on your screen, you will see a couple of different tabs. One of them is called Chat. In that chat box, we'll be entering a little bit of information there over the course and towards the end of the presentation. But there's also a tab that's called Questions, with an arrow pointing at the questions. And if you click on that, that'll pop down a questions tab. And if you have any questions for our presenter that he'll answer at the end of the presentation, you can enter them there. And I'll be viewing all of your questions and fielding them to our speaker. So just so you're aware of those things. Once again, this is being recorded, and if you do need to leave early, you will receive a link to this recording, which, by the way, you can also share with anyone else you think might be interested in this. Today's presentation, I would be remiss in not thanking the sponsors of this presentation. It's possible you heard about today's presentation from us directly at Worldwide Quest. It's possible also through one of our partnering Canadian University Alumni Associations. So you might recognize one of these, or maybe two or more. All of these partners are, in fact, sponsoring the presentation around which today's presentation is built, the tour rather, around which today's presentation is built. So we thank our partners for sharing news about this educational opportunity. I can tell you a little bit more about us. As I said, we're a small travel company based in Toronto. We've been around since 1970, and our specialty really is in small group experiential and educational travel. We go to all 7 continents, and we feature what I'll say is congenial and informative leaders, experts on a number of different topics, nature, culture, or the arts. This year's a happy group of travelers in the Magdalen Islands. Today's presentation is by one of the leaders on one of our expeditions coming up, the Epic Antarctica with the Falklands and South Georgia, coming up later this fall. I'll tell you more about that towards the end of the presentation, but suffice to say, it's we think it lives up to the name Epic. This is a grand circle expedition visiting 3 key sites in the Antarctic region. And as I said, one of the features of our trips really is the expertise of our leaders. And on this particular expedition, being as it is a larger expedition, we have in fact 3 study leaders drawn from those participating Canadian universities. These are study leaders Philippe Tortell, who's today's speaker, an oceanographer from alumni UBC, Philippe Tortell, also an oceanographer of sorts from McGill, and Brent St. Clair, a biologist from University of Western Ontario. All of these 3 speakers, who in fact have been presenting a series of Antarctica talks we've been sharing with you, will be aboard with their own series of talks, forming a beautiful whole as we travel through the Antarctic region, allowing you to understand the context for the region and see much more and understand much more. Today's speaker is Philippe Tortell, oceanographer with alumni with University of British Columbia. Philippe, in fact, was a study leader with us on our 2022 expedition. And I'd like to welcome Philippe to join us now. Philippe, are you there? Hello, welcome, Philippe. We don't hear you, Philippe, though.
Okay, now I think I got it. So you can see me, I think, and you can hear me?
Yes, we can, certainly.
Okay. All right, now the final step in all of this, I think, is to share my screen. Let's see. How about this? Are you seeing that, Justin?
Yes. Perfect, thank you.
Okay, super. So I think we're all set here. Thanks everyone for making some time to join us. I'm quite pleased to be with you. I'll be pleased also. Whoa, wait a minute. I'm just now seeing myself all of a sudden. That's weird. Why is that? Okay, you're still seeing my screen, right?
Yes.
Justin? Yeah, okay. So it's, as you know, it's a little bit strange to stare at your own screen and be in a bit of a virtual vacuum, but Justin, I'll rely on you to let me know if things go haywire or you stop seeing the screen or whatever. Just please pipe up and let me know. Otherwise, I'll assume that everything is fine. So as I said, I'm quite pleased to be with you today to share a little bit of information about the kinds of oceanographic environments and ecosystems that one would encounter on this trip. As it says here, it's the invisible world of Antarctica's marine ecosystems. And I say invisible because there are many visible things in the Antarctic, like this whale that's shown here and the seabirds and other marine mammals. But what I am going to try to get across to you today in a relatively short presentation is that it's the things that we can't see, or at least that we can't see quite obviously, that are actually pretty foundational to shaping the way that these ecosystems actually work. So I'll give you a very quick run through from the oceanographer's perspective about the different elements of the physics, the chemistry, and the biology that conspire together to make the Antarctic such an interesting place. And if there's time permitting, I'm going to share a little bit of the experiences that we had in 2022 when I participated in the last iteration of this. Let's see. So hopefully I can change screens. There we go. So here's a picture taken from an Antarctic ecosystem, not on that particular trip in 2022, but previously. And what I like about this is that you can see the different elements that come together to influence the nature of Antarctic marine waters. So what do we see? We see the water itself, but we see ice, we see mountains and continents, and we see the atmosphere. And all of these things, as it turns out, really play an important role in shaping the structure and the function of Antarctic marine ecosystems. And the question that we're trying to understand is, how is it that ocean physics, chemistry, and biology all come together to shape these marine ecosystems? And the analogy that I like to start out with to really put some context around this is a cake. Here's a chocolate layer cake, and you'll see that this is actually a fairly important and interesting and useful analogy for the way that marine systems work. And we can think about that cake as a parcel of upper ocean, maybe a few hundred or even a few thousand meters deep. And at the top of that cake, at the top of that layer of icing, we have an atmosphere, or we have ice. These are things that sit over top of that very upper level of the ocean. And that's where we have the exchange of heat and light, exchange of gases, nutrients, and fresh water are largely all interacting with the surface ocean across that top layer. At the bottom of the cake, to continue the analogy, we have an interaction with the seafloor. And that's where we have other sources of nutrients coming in. Along the side of that cake, we have the continents, we have glaciers, and those are exchanging sediment, ice, nutrients, and additional fresh water. And then within that, very importantly, as you can see here through this diagram of the different layers of the cake, we have the exchange of different water masses across these layers by a process known as vertical mixing. So we tend to focus our attention as humans on that very upper layer, that little skinny layer of frosting on the top of the cake. But I'm going to try to convince you that actually understanding how those layers of water move vertically up and down in the water column plays a really important role in controlling the way that Antarctic systems work. I'm going to give you another food analogy. Maybe it's lunchtime where you are, maybe this is going to make you hungry, but it's an apt analogy, and I'm going to talk about ocean mixing and what we call stratification. And the concept here is that the density of seawater, which is basically the amount of weight per volume, so kilograms per liter, for example, is controlled by its temperature and its salt content, or what we would call the salinity. The upper ocean is low in density, so it's a relatively lower mass per unit volume than the deep ocean, which is a higher density. And it's very much like the situation of an oil and vinegar dressing here, where the oil is a lower density liquid than the aqueous or the water-based vinegar, and it therefore it has a tendency to float on top because of its lower density. And in the ocean, that upper layer is the layer where we receive sunlight. So it's the layer where photosynthesis can happen, but the nutrients tend to accumulate in the bottom of the ocean. Organisms grow up in the surface through photosynthesis, they produce organic material, and that material is constantly settling down into those lower layers of the ocean. So the upper water has sunlight, but typically is poor in nutrients, whereas the deep ocean is dark, hence the analogy here I think works particularly well, but it's rather rich in nutrients. If we take that bottle and we shake it up, we introduce energy into it, what you can see is we have this emulsion, and that's very much a good analogy for what happens in the ocean when we introduce energy in the form of wind and wave energy. We can mix the ocean and we can introduce some of those nutrients from the deep layer up into the sunlit surface, creating conditions that are favorable for photosynthesis. And so this is this process of mixing, which is very, very important in the ocean. And here's just a quick little video showing what we do as oceanographers to understand something about the vertical properties down through these layers of ocean, or cake if you want to keep thinking about it that way. This is an instrument that we would send down that's equipped with sensors for temperature and salinity, and it allows us to also collect samples of seawater. And I don't know how easy it is for you to see, but these bottles have a little each of these bottles has a little round hole in the middle of it. These are pteropods, by the way, that you're starting to see zooming across your screen. And in a moment, you're going to see one of these bottles close. And these bottles are triggered at a particular depth from inside the computer, inside the ship on a computer, and it allows us to capture water at a particular depth to bring it back onto the board to capture water properties and make various measurements. I think it's coming up here in a second. Not that exciting, I guess, but let's see if we get there. I'm going to look at the pteropods in the meantime. Oh, there we go. I just zoomed to that. Okay, so watch this bottle here near the cable. It closes, and then the instrument package goes back up. So after many, many years and decades of making these different measurements of temperature and salinity in the ocean, we can characterize what we call different water masses on the basis of their temperature and salinity. So this is what we would call a section. Imagine this is like a slice of that cake. We're looking here on the left, the south. The black is basically the ocean floor. We have the Antarctic continent, and then we get mid-Atlantic, mid-oceanic ridges, and so on. And these different layers here with the arrows represent different water masses, again, characterized by very distinctive temperature and salinity. And we can see that these water masses tend to exist in some isolation because they're separated by these different layers of density. And here's just a slightly more scientific, if you will, version of that, but now the colors. So I'm still showing you a section, so I should guess I could explain a little bit better. When we look from the top to the bottom here along the vertical, that's ocean depth. So this top layer is basically the surface ocean, and I'm showing you down to about 300 or 400 meters below the surface. As we go from the left to the right, we're going from towards the Antarctic. In the middle here, this is the equator, and on the right, we have the North Atlantic. And the color represents the concentration of nutrients, nitrogen in this particular case, or nitrate. The blues and the purples represent very low concentrations, so very nutrient poor surface waters. And when we get down here to the Antarctic, you see we've got a lot more greens and yellows, and this is an indication of the fact that we do have in these higher latitude regions, certainly at some times of the year, we have lower stratification or higher mixing, like we're shaking up that oil and vinegar and introducing some of those deeper waters closer to the surface, and therefore introducing some nutrients. So this process of mixing that introduces high nutrients in the surface is really a key feature of why, or key characteristic that explains why Antarctic ecosystems are so productive. So again, in the low latitudes, we've got these highly stratified systems, and where we get more vertical mixing and lower stratification in the Antarctic, we bring those nutrients up. And a lot of this is related to the fact that waters are simply colder. As we go to higher latitudes, closer to the poles, north and south, the water temperature decreases. This is a map of sea surface temperature measured by satellites. The reds and the yellows and the oranges represent higher colors. And as we go down towards the Antarctic, not surprisingly, the temperature decreases. And what that decrease in temperature does is it increases the density of those surface waters, and therefore decreases the density difference or the stratification between the upper ocean and the deeper ocean. And that weaker stratification makes it easier for mixing to churn up those nutrients into the surface. Another important thing when we talk about the atmospheric imprint is the role of atmospheric dust transport. So this is a model generated from NASA observations, satellite observations, and what I want you to focus on in particular, so this is Antarctica here in white, of course. To the left of the Antarctic Peninsula is the bottom tip of South America. You can see some brownish area here in eastern southern portion of South America. Those are the very dry areas of Argentina. And this little brown sort of tail that you can see spinning off of South America is a signature of atmospheric dust that's picked up off of the continent and transported for long distances over the ocean. That's important in our context because that dust turns out to be a very significant source of iron, which is a micronutrient that's really quite important for biological production in all ocean regions, but particularly in the Antarctic. And then finally, we can talk about light. Light is another thing that comes from the top. Obviously, the sun overhead is the source of light that fuels photosynthesis. That is highest in the surface, and as you can see in this image, is decreased very rapidly with depth. As water molecules scatter, the light and other compounds dissolved in water absorb photons and therefore reduce the light. And sea ice in particular can play a significant role in reducing the amount of light that penetrates into the ocean. When we've got very thick ice cover, as you can sort of see from this picture, that ice is going to rapidly attenuate water by, rapidly attenuate light by essentially reflecting a lot of that light back out into space. And so this is just a animation that I made of sea ice cover. This is years worth of daily sea ice data. The white is sea ice, and you can see the date here ticking away. I think this is just a few months. So starting in October, we're going to be here on the Western Antarctic Peninsula. You can see that sea ice eventually is going to disappear, creating open water that's favorable for phytoplankton growth. All right, so putting that all together, we understand that sea ice cover, light, temperature, nutrients, all of these things are really important to structuring biological productivity. And we can get a sense of how that works if we look at a comparison of sea surface temperature, which is shown here on the light from satellites, on the left, excuse me, from satellites, and then a map of chlorophyll concentrations on the right. These chlorophyll is a pigment that's used to generate energy from light, to essentially harvest light for photosynthesis. It is present in all photosynthetic organisms. And by looking at the color of the ocean, actually looking at the different wavelengths of light that are emitted from the ocean, we can make fairly good estimates of the chlorophyll concentration. So in this map here on the right, the colors that are very blue and almost purple represent areas of very, very low concentrations of chlorophyll. Now, interestingly, these are the areas where the surface ocean is relatively warm. Putting the ideas together, hopefully you begin to understand that the areas of the ocean that are very warm, like these tropical places like Hawaii or Bermuda or whatever, these are places with strong stratification, and those nutrients are largely stuck down in those deeper waters. And therefore the chlorophyll and the phytoplankton biomass is low. As we go into these colder areas, however, the stratification is weaker. It's easier to mix those nutrients up, and correspondingly we see high concentrations of chlorophyll, as shown here in the greens and the reds and yellows and so on. And then I don't know if you remember, but I showed you this tail of dust coming off of the eastern southern portion of South America. Here, if you follow along this dotted line labeled with a 3, there's a very interesting plume of high chlorophyll that turns out to track that dust plume pretty nicely. So that's a signature of an iron fertilization, a natural iron fertilization of biological productivity in the Southern Ocean. And when we talk about biological productivity at a microscopic scale, there's a variety of different organisms, but in the Antarctic, primarily 2 groups. One is a haptophyte. It's actually a colonial organism called Phaeocystus antarctica. Each of these little bigger round things is a colony of Phaeocystus antarctica. Each of the individual little dots would be one cell, one individual cell of Phaeocystus antarctica. There are about 5 microns in diameter. A micron is a millionth of a meter. And over on the right, I'm showing you an image from a scanning electron micrograph of diatoms. These are a fairly diverse, a very, very beautiful group of phytoplankton, which is notable for the fact that cells have a cell wall made, a cell covering, I should say, made out of opal, which is basically glass, it's polymer of silicon dioxide. So these are the kinds of the organisms that underlie the biological productivity and drive photosynthesis, and therefore support the rest of the food chain. And the next step in the food chain is euphausids, or krill. These are organisms that basically large shrimp-like organisms that feed directly on the phytoplankton. And the krill themselves can be consumed directly by baleen feeders like whales, also by penguins, seabirds, and other things. And this is really important because the food chain in the Antarctic ecosystem is dominated by relatively large phytoplankton like diatoms, which can be fed on directly by krill, which in turn can be fed directly on by seabirds, mammals, and so on. And that's important because it is what we would call a short food chain. In other words, it's only 3 steps from the beginning to the end. So phytoplankton, krill, whale. And that short food chain means that a lot of energy can be efficiently passed on from one trophic level to the next. In ecology, there's something that we call the 10% rule, which is to say that only about 10% of the energy is passed down, or passed up, I should say, from one trophic level to the next. So if we had, let's say, 100 units of phytoplankton biomass, we would expect only about 10 units of krill biomass, because 90% of that, or 90 units of the biomass, or the carbon and energy, is burned up through respiration and a little bit of heat and so on that's lost. And if we had 10 units of krill biomass, we would only expect 1 unit, or 10%, of whale biomass. If we had to have a couple of extra steps in here, if we had maybe 2 groups of phytoplankton and the krill, and then something else eating the krill, and then something else eating the something else, and then finally to the whales, each of those times that we introduce another step in the ecosystem, we're basically losing about 90% of the carbon and the energy. So just to repeat, the fact that we've got these very short food chains in the Antarctic means that we're very efficiently, or as efficiently as possible, transferring energy from the base of the food chain up to the top consumers. And here's just a nice picture of some whales, just giving you a friendly wave here. Just to say a few words about climate change in the context of this. So I've described very, very briefly the impacts of light and temperature and mixing and all that kind of stuff on nutrient distributions and phytoplankton. This, of course, is all playing out against a rapidly changing climate. This is an image of temperature change. This is an anomaly. So we take kind of a 50-year time record and we normalize everything to some average, and we're just comparing things over this 50 years relative to some long-term average. So the values that are shown in blues represent an actual decrease in temperature relative to some average, and the reds are a positive warming signal. And I'm sure you'll all know that climate change is leading to a strong warming of the Northern Hemisphere and the Arctic in particular, as shown here by the red. What's not so well known by a lot of people is that there's a strong spatial variability to the signal. So unlike these areas in red, actually much of the Southern Ocean at the moment is seeing a slight cooling trend. The big exception is the area here around the West Antarctic Peninsula, which is showing a high rate of warming. And that's the region where this particular trip is going to be going. So indeed, there is a significant exception for warming on the West Antarctic Peninsula. And similarly, if we look at some sea ice trends, this is just the total volume of sea ice expressed as an anomaly relative to, at this point, I guess it's about a 40-year data set. The most recent ones I've gone are only to 2020 or so still a few years out of date, but nonetheless, you'll get the idea. A strong trend towards decreasing sea ice cover in the Arctic, and the Antarctic, notwithstanding some big bumps and wiggles, and I don't know exactly what's happened here from 2014 to 2017, but the point I'm making is that there is still a contrast in global, rather regional sea ice cover in Antarctica, which is showing perhaps a slight positive trend or maybe no trend, versus the strong decrease we're seeing in Antarctic sea ice. The point here being is that the Antarctic and the Arctic, both these high latitude polar regions, do seem to be experiencing fairly different changes to, or different responses to global climate change and global warming. And that is associated in the region that we'll be traveling to in the West Antarctic Peninsula with some changes in the distribution of phytoplankton. Again, looking at chlorophyll distributions, these are anomaly plots. So the red values here in the southern West Antarctic Peninsula indicate a region where we're seeing an increase in phytoplankton abundance based on chlorophyll, whereas in the northern Antarctic Peninsula, I think where this trip will be spending more of its time, there is a trend towards a decrease in phytoplankton abundance. That has been linked to the warming of the surface ocean with the reduced enhanced stratification and reduced mixing and the reduced input of nutrients, as we've discussed. Justin, how am I doing for time? How much time have I got?
You have a good 10 minutes.
Okay, so what I'm going to do now is sort of, having given you a little bit of a background, I'm going to give you some examples of how we are going to explore some of these things on the cruise through my own participation and some of the activities that we'll do together and some of the instruments that I will bring on board. And remember here, the theme is making the invisible visible. When we go down there, you're going to see whales and birds and other marine mammals and all this kind of stuff. And that's beautiful. That's really the highlight of the trip. But my interest in participating in this is to help people on board understand that it's those things that are invisible to the naked eye that are actually really exerting a first order control on all the stuff that we can see. So I call this science education and outreach on the Magellan Explorer. And just for context, I want to say, as an oceanographer who's been working in this field for almost 30 years, I do have a lot of experience working all over the oceans on research vessels of all kinds. And only in the last few years has it come to my attention that there are a fairly significant number of polar tour operators who are looking ways to provide interesting, interesting experiences for quote unquote high-end customers. And I say high-end customers here in the sense of focusing on individuals who are really interested in learning and understanding the environments in which they are traveling. So people who are fairly discerning and not just looking for, kind of a quick fix, a few pictures, but really interested in immersing themselves into an experiential learning opportunity. So these passenger expeditions provide a platform for environmental monitoring, as it turns out, for some pretty important variables in critical polar regions that are changing rapidly. And it is the case, certainly, that increasing number of tour operators are making significant investments into science. There's a French group that I've worked with just a few months ago, actually, that has built a bespoke state-of-the-art laboratory right into their passenger vessel. Now, the Magellan Explorer doesn't have those capabilities, but what we've done with them through our partnership starting a couple of years ago is that I got them to install a new seawater line in the library, which is on one of the upper decks. So they plumbed in a line all the way down from the engine intake, and they fed it all the way up into the library so that we could have a continuous flow of seawater going up into that upper laboratory. And so this is what I did. I showed up on the day of the embarkation with everybody else, and I scrambled around with the ship's engineers, and we did an installation of this seawater monitoring system that consists of a few different devices. First is a thermosalinograph, so that measures temperature and salinity. And I put an oxygen sensor on there, and I also put in what's called a total tension device, or GTD, gas tension device. And that measures total gas pressure in the seawater, which can be used to derive the nitrogen concentration. The details don't really matter, but what I want to say is that this seawater line that I showed you was feeding in directly to these different sensors, giving us essentially a real-time, very high-frequency set of observations around these key parameters. Temperature, salinity, and oxygen are the ones I'm going to focus on. So I'm going to show you some of the results that we got from this trip and put it back into the context that we've created over the last few minutes. I just mentioned that this was turned out to be fairly popular. It was a point of connection with the passengers. We had a number of different little informal talks, and people were invited to come up anytime actually they want and look at the results. People really got quite interested in understanding what was going on. And the data were available in real time. So there was a little screen set up, and you could watch the data coming in real time, and you could go there and say, well, what's the sea surface temperature? What's the salinity or the oxygen saturation? And I'm just going to walk you very, very quickly through this. Maybe let's just focus on the temperature. This is the top plot. I started measuring somewhere south of South America, and you can see all kinds of bumps and wiggles, but the ones that I want to highlight are the two here and here at around November 3rd or so and November 15th, which is when we're going down into the Antarctic and then back up towards South America. We cross what's called the Antarctic convergence, or I prefer to call it the Antarctic polar front. And this is a strong, strong region where temperature changes very, very rapidly. There's also salinity changes and so on. And that's really an oceanographic demarcation that tells us when we've crossed over into the Antarctic zone. And I'll just mention that the oxygen here is represented as a percentage of atmospheric saturation. So if the value here is 0, as shown by this dotted line, it means that the concentration of oxygen in the water is exactly what we would expect based on exchange of gases between the atmosphere and the surface ocean. When the values are greater than 0, it implies that there's an additional source of oxygen in the water column, which is attributable to photosynthesis largely. There's a few other things too, but we'll just keep it simple and say values of oxygen saturation greater than 0 give us an indication that we have a lot of photosynthesis in the water column. Conversely, areas where we have some values of oxygen lower than 0, oxygen saturation lower than 0, implies that we have a little bit of excess respiration going on. And actually, in fact, this really reflects just mixing of some of these deeper waters up into the surface. So we get a lot of information just by measuring temperature, salinity, and oxygen. And I'll just mention that we use this on the way back up. We created a little competition and we asked people to guess when it was that they thought we had crossed the polar front. So people would go in and we described a little bit about what the polar front is, and they had to guess at what time we were going to cross, and whoever got closest won. I think it was a pair of penguin socks, if I'm not mistaken, Justin, something like that. Anyway, a fun little competition. All right, so this is just a map now showing you the same data. Probably easiest just to focus on temperature on the left, and you can see this really strong gradient in temperature as we cross the polar front. The blue, green values are the lower ones, and as we get to the yellow, we have the warmer waters further to the north. And these are the oxygen data that I showed you, and actually you can see some interesting patterns. Again, focusing here on the left, at the very bottom of South America, shown there, the Patagonia, you can see some gray, some sort of thin gray lines. That's the continental shelf. That's the bathymetry or the bottom depth of the ocean. You see a little spike of yellow perhaps down at the bottom there, and that just shows you high productivity that's associated with the continental shelf and the input from nutrients and some freshwater, the input from the continental shelf and rivers that's introducing some nutrients. Now this feature over here to the right, closer to the South Georgia island, is kind of interesting. Let me just jump ahead and show you an image of chlorophyll that's measured from a 20-year average from the satellites. And what I find interesting about this is that the area of high oxygen, remember this is high biological productivity, 10% supersaturation in oxygen, kind of lines up pretty nicely with what we're seeing from the 20-year average of chlorophyll, where again, the yellow values are the higher ones. So these areas where the satellite is picking up higher phytoplankton biomass shows up really pretty nicely in our shipboard oxygen data. And, you know, spoiler alert, this is related to inputs of iron that are coming in off the continental shelf and inputs as well associated with the presence of South Georgia Island. There's more to it than that, but that's certainly part of the story. And then finally, excuse me, in addition to the measurements and the instruments that we brought, we participated in a wonderful citizen science program that's run by the crew of Antarctica 21. This is us on a boat, a little Zodiac. We collected some water samples that we filtered, and we made some chlorophyll measurements, and we also collected water that was concentrated in this way that we used back on the ship to do a little activity with the passengers. We did this during a day when the weather was poor and outings weren't possible. We took the water, we made some slides, we projected it in the main lounge area. And in addition to that, so that what's shown here on the screen are some beautiful centric and pennate diatoms. The things that sort of look all stacked together, those are individual cells that are forming a particular chain, and these round things are individual cells. So these are diatoms that are associated with the sea ice, which is where we collected the sample at the bottom of some sea ice. And then in addition, we brought along these things called fold scopes. Maybe some of you have heard of them. They're basically $1 or $2 paper microscopes. They were developed by someone at Stanford for use initially in the developing world to help with identification of pathogens and so on. We brought along about 80 or 90 of these. We gave them out to just about everyone who wanted one. And people had an opportunity to make these microscopes and look at some samples underneath their little paper microscope. And here's one of the passengers who was particularly interested in that. And the image that you can see on the right is one of my favorites because it shows someone who's looking out the window of the ship towards an iceberg, but actually they're not looking at the iceberg, they're looking at the microscope. So it just shows you that we were able to capture their attention and make that invisible suddenly visible. And you're not going to be able to hear the audio on this, but this is Aldo. Don't know if you could read lips or not, but you'll get a sense pretty quickly that he was fairly enthusiastic about the activity, talking about one of the best things I've ever done, I think is what he says here. So he's got the fold scope, and let's just see. Hang on in a second. You'll see him. And what does he say here? Just kind of talking about his experiences and how it sort of makes all of this come to life for him. Again, I wish I could share my audio, but I think you're getting the sense. And then he was a particularly engaged person, so you can see how excited he is about his foldscope. Microscope is where he says there. Talking about the other microscope that we had, and on the screen, as you can see, as I've shown you, this is what we had projected up there. And there's that little other microscope that we had brought along. So there you go. And so that's what I wanted to get across. If we have time, of course, I'm happy for any questions and discussion. Thank you.
Well, great. Thank you so much, Philippe. Very interesting as always. Before we go to questions, I did want to mention, you may have noticed, those who are attending today, we did have a book prize as a door prize giveaway, but you'll have to stick around till the very end of this presentation to receive that. So we do have some questions here. We have one from Sharon, who is asking, she says, you talk about phytoplankton, but what kind of zooplankton do you see?
Yeah, so the classic zooplankton are krill, as I talked about, the euphausia. They're present in very, very high abundance. There's another group of gelatinous phytoplankton called salps. They can also be present in significant concentrations. When you get big blooms of salps, that has fairly significantly negative impacts because they are these sort of gelatinous things, and they're not very good food for birds or marine mammals. We also get organisms, little crustaceans called copepods, tend to be much, much smaller than krill. And I would say between those three, those are really the dominant zooplankton groups: krill, copepods, and salps.
Excellent. Just looking for other questions here. Zelma asked a question, which I think you might have answered ultimately, but initially she was asking, why is there not the same level of ocean vertical mixing in the Arctic as in the Antarctic?
I didn't answer it directly. That's a really, really interesting question. And the reason has not to do with temperature, because the Arctic, surface Arctic Ocean is just as cold as the Antarctic. What's different, though, in the Arctic is that there are many, many, many rivers, a lot of fresh water that drains from the continent around the Arctic, so the Eurasian continent and the North American continent. And all of those rivers put a lot of low salt content or low salinity water in the surface Arctic. And it's that low salinity water that creates very low density that prevents the mixing. And in the Antarctic, we don't have rivers. We just don't have the same source of fresh water, and therefore the density is just higher.
That's a great answer. Very good. Thank you, Zelma, for asking that one. I'll wait a couple of moments to see if there's any further questions. I did just want to remind everyone who is here that, of course, the expedition that Philippe and I have been referring to is our Epic Antarctica expedition that will be taking place once again this October and November. This is a full circle voyage, which takes us from South America through to the Falkland Islands, then through the South Georgia, then onto the Antarctic continent proper, Antarctica, before we circle back to the southern tip of South America. Of course, Philippe, you alluded to South Georgia being a hotbed of productivity, and we have a chance to see firsthand when we get to South Georgia the effects of that ocean productivity and all those multiple factors that you've been talking about that converge on that region in particular. So I do have another question here. Yeah, that's an interesting question. I'm not sure if I understand. Oh, I think I think I might understand this. From Andrew. Andrew has asked, it's an interesting question, and I think he's referring to flights, maybe air travel. We know that during COVID with lockdowns, there was a dramatic reduction in CO2 production coming from flights. I'm not sure how much effect that had. I guess the question is, did that, whatever that reduction was, have any effects on the Antarctic?
No, I know a little bit about that. It was observable. I don't think it was massive, and it was also short-lived. So I think the answer is probably not any demonstrable effects on the Antarctic.
Interesting. I have another question now from Blaise, who was asking about a similar question. Let me see here. 10% offsets. That's a great question. Blaise is asking about carbon offsets for this trip. I don't have the information at my fingertips, but our partners on Magellan Explorer do have a whole statement about sustainability, which I can make available to you. I don't have that right now. Dale is asking another question. This trip is quite early in the Antarctic spring. How does that affect the range and numbers of animal life seen on the trip? Well, Dale, I could just answer that directly, that all the wildlife that you would expect is there. The main thing about traveling in the springtime is that it's early in the courtship time of a lot of the wildlife, like the bird life. The penguins, for example, are just starting to nest. It's also in the courtship time of things such as the southern elephant seal, those huge hulking seals, the males of which battle each other for supremacy of beaches. So that's the time when you see those very specific activities. The springtime also means that the colonies, birds and seal colonies, are also relatively clean, if you will. They are devoid of guano, large quantities of guano. So they tend to smell fresher. As well, in Antarctica proper, there is still a nice fresh layer of snow, which, as penguins are returning, it's a very clean look. It hasn't been, it hasn't received the guano inputs that the penguins sort of deposit over the course of the season. So basically, it's a much fresher, cleaner time to be in that season. Otherwise, wildlife, by and large, is the same. So that's there would be maybe fewer whales seen in the early season. They tend to build up towards the latter part of the Antarctic summer. But we have seen all of the full suite of wildlife that is possible to see during that time. I'm not sure if there's any other questions here. I did just want to mention once again that Philippe will be a presenter with our Epic Antarctica with the Falklands and South Georgia expedition coming up this fall. A very special expedition limited to 88 travelers only aboard this very nice small ship expedition cruise. It's a very high resource and study leader ratio to traveler. So we hope you'll consider joining us. I'll share some more information about that expedition afterwards. You will be receiving a recording to this presentation. And I did also want to just take a moment here to go to our prize. I mentioned there would be a prize for participants. I'm just going to get the name that my colleague Fishwa has drawn from the list of live attendees, and the recipient of this wonderful book, Antarctic Atlas, New Maps and Graphics that will tell the story of the continent. We'll call it generally a very different sort of atlas, which is on our reading list, our recommended readings for this exhibition. The winner is Robert Dewar. Robert, we'll be in touch with you to make sure we have your mailing address. Congratulations to you, Robert. I did want to thank everybody for attending this. I would like once again to thank Philippe for a super informative presentation, which is really just a taster of a series of presentations Philippe will be doing for us aboard. Getting us much further in depth on the topic of ocean productivity and sustainability. Thank you very much, everybody, for attending today, and we'll look forward to being in touch with you, perhaps seeing you at a future event or perhaps aboard this expedition. Thank you.
All right.